My name is Davide Michieletto. I'm a reader in statistical physics and the group leader of the Topologically Active Polymers Lab. Welcome to my exp. So deoxyribonucleic acid, or DNA for short, is the blueprint of life. So it contains all the genetic information required for complex organisms to be alive and to sustain life. Now, we've been knowing since the 1800s with Mendel and Charles Darwin as well, to some extent, that there are some genetic traits in us that can be preserved and passed to future generations. But it was not until the 1950s, 1940s, that people started to ask the question of where are those genetic traits stored? So, and originally they were thought to be stored in proteins until the 1940s, and then slowly people started to realize that perhaps they were stored in this deoxyribonucleic acid, or DNA. And that led slowly to several teams trying to race each other to discover the structure of DNA. And since 1953, since the Watson-Crick paper, we now know that DNA is made as a double helix, so it's two strands of nucleotides, and these nucleotides can be ATCG, or adenine, thymine, cytosine, and guanine, which are placed one after the other, giving unique sequences along our genome, along our DNA, and they encode the genes, the blocks of information that are required to encode genetic traits. In this image, you see the extreme level of packaging the DNA is under inside a living cell. So in this E. coli, Escherichia coli is a bacterium, so it's a single cell and contains its whole genome in that cell. And in this particular picture, what you see is that the DNA has exploded. So the DNA, the bacterium has exploded out. And what you should imagine is that all that DNA is compacted, is confined within that little cell that was originally the live bacterium. And to me, one of the things that, the first thing that everyone should notice is how extreme and unimaginable is the level of compaction that DNA is experiencing inside the bacterium. So DNA, this picture really tells a lot about the fact that DNA is not simply this abstract object that contains genes and genetic information. It's really a physical object, almost like a physical rope that you have to stuff into a bag, let's say, into a membrane to make a living thing, to make a living organism. So DNA in bacteria is stored so-called naked, so it doesn't actually, is not wrapped around, it is packaged using proteins, but is not packed into chromatin. In eukaryotes, which eukaryotes means higher order organisms essentially, so eukaryotes are ours, we are eukaryotes in the sense that we have a cell and it contains a nucleus, and the nucleus of the cell contains the genome. So in eukaryotes, DNA is packaged into what you see in this image. So it's packaged, it is wrapped around histones, which are proteins, and these histones, so the complex of DNA and histone is called the nucleosome, and it's packed together again into a fiber, and the fiber is called a chromatin, and this chromatin is then itself folded into three-dimensional shapes into the cell nucleus. And what is really interesting and what's really I'm really passionate about this, is that in the past 10 years, people are starting to discover that the shape, the way chromatin is folded in the nucleus affects the way genes are expressed. So in eukaryotes, in plants, in animals, in humans, there is about 20,000 genes. Obviously, it depends on species, depends on whether it's a plant or an animal, but it's about that order of magnitude of genes, 10,000, 20,000 genes. But not all of them are active all at the same time, and this is an important point to make, in the sense that DNA, and in particular chromatin, is not a passive book. So the cells in a complex organism like us, they need to express different genes because they need to execute different functions. For instance, a muscle cell is different from an eye cell or a kidney cell or a liver cell. So they all have to express different proteins. And to express different proteins, they have to have different genes active, different patterns of genes that are active. And this is part of to understand what determines the patterns of genes. We have to look at genome organization, how the genome, how the DNA and chromatin is organized inside the cell nucleus. And this is what many groups are doing, and we are also contributing a little bit in this field. The DNA and the genome responds to environmental clues. So, for instance, in plants, I was discussing it quite recently with a colleague in plant science. Plants have a different immune response, let's say, than us. So what happens is that when plants are attacked by pathogens, cells can sense the presence of these pathogens using sensing proteins. And these sensing proteins act to change the transcription pattern itself. So the DNA transcription pattern changes depending on whether the cell is being attacked. And when the cell senses this attack by the pathogen, by the attacker, it starts to create defense proteins. And these defense proteins then defend the cell against the pathogen, or they could induce cell death. So the cell death, so for instance, when you see spots, yellow spots on green leaves, sometimes it's because the cells decide, that spot of cells decide to die to save the rest of the plant. Then my question was, why doesn't the cell always produce these defense proteins against pathogens? And this is because it will be too energetically costly. And so the plant, so if it produced always these proteins to defend itself against pathogens, it would affect the growth. It will reduce the growth of the plant. The plant has to decide whether to grow or to defend itself. And so this is also another example of how active is our genome and of complex organisms. It's an active book that changes in response to what happens nearby. And so this is the challenge, I think, the big challenge in the field of genome organization is to try to understand not only how a static snapshot of how it is organized inside the cell in a given time, but how it changes over time. So then the dynamics of the genome inside the cell, and how, in some sense, also how the entanglements and the structure of this chromatin, how the folding of this chromatin affects the behavior, the response to attacks, or the changes in gene expression when the cell goes from healthy to diseased, for instance. The history of DNA discovery has been a very tumultuous one, I have to say. So clearly in the 1930s, 40s, and then 50s, there was a lot of effort and a lot of teams that were interested in resolving protein structures. And then some of the teams also started to use the same techniques, so X-ray crystallography, to resolve the structure of DNA. And there were several teams in the UK, so there was the Cambridge teams, and there were Watson and Crick, but there was Rosalind Franklin with Wilkins in London. There was Max Perutz as well in Cambridge, and Linus Pauling in Caltech with Max Delbruck. We're all trying to get to understand the structure of DNA, and it felt, from what you read in the books, is that it really felt like a race to who got there first. And I remember reading that when Linus Pauling came up with the first prediction of the DNA structure, which was wrong, was he predicted a triple helix with some also other incorrect predictions about where the phosphates were. Bragg here in the UK was quite upset and started to kind of put pressure on the researchers here in the UK to get there first, to try to get to the correct structure first. And this is how then Watson and Crick kind of got hold of the DNA structure, crystal structure that was generated by Rosalind Franklin in London. And that was still unpublished at that time, and Watson and Crick got hold of this picture and managed to come up with the correct structure. So that, to me, whenever I read this story, it always feels very current. It feels like it's something that could happen even now, in the sense that academia often is idealized from the outside, but in truth, what happens inside is often feels like a race. Who's the first team to get to a certain answer, a certain prediction? It's not always about a healthy competition, and sometimes it seems that it is done to fuel the ego of some people rather than really genuine passion for discovery and for science. One misconception that I would like to fight is the fact that DNA is not only an abstract object that contains genes and genetic information, it's also a physical structure. It's a double helix with the Watson-Crick base pair rules, and the fact that it has this structure makes it an active participant of deciding which genes are expressed, and therefore how cells behave, and therefore how we are built as complex organisms. So it's a very important concept that I think is not understood in the general public, that is really a physical object that contributes to its own function.